All right, you guys, so this is our first little tutorial that we're going to have for our soft closure. So just so you know, I am just doing the homeostasis lab book, and I am currently on this page. It's page 6, and it looks like this. Okay, so that's the notes page we're going to do through today, is this active transport. All right, guys, so the biggest thing to understand is it is definitely transporting material in and out of the cell, but there's one major detail, is you got to have ATP. You got to have a mitochondria to make lots and lots of energy. So here's our cell membrane, better known as the phospholipid bilayer. So here's your phosphates, there's your fatty acids, your lipids. Just a little reminder, here's your proteins. Now a substrate is just a larger substance that comes in and out of the cell. Now something unique about the substrate is that only certain proteins can bring in certain substrates, kind of like a lock and a key. So this substrate is unique to this protein. It can come in and enter the cell as long as it fits the right protein. So just something to keep in mind, that only certain proteins can bring in certain molecules, certain substrates. All right, so there's the outside of the cell, here's the inside of the cell, and here's the proteins bringing it in. And once again, if it's active transport, it requires energy. Passive transport, no energy. All right. So we're finally, when it comes to active transport, remember the example that I gave you of if you were in a kayak and you're going with the current, then you can only go from a high to a low. You can't use any energy. You don't have any paddles. You don't have a motor, so you can only go down current. But with active transport, now we just put a motor on the back of our kayak, or now we have paddles. So now we can use energy. So this definition of active transport is just like passive transport, except it uses energy. So the transport of molecules from areas of high concentration high to low concentration, or it can go low to high with the use of energy. So that's unique. That's different than passive transport. So the cell's energy is the ATP made in the mitochondria. So if you look here, when I originally gave you the example of being in a kayak going down current, so active transport can definitely go from a high to a low concentration. That hasn't changed. So it can go with the current, or if you have paddles, you can go against the current. So you can go from a low concentration to a high concentration, which you couldn't do with passive transport. So that's different. So here's a picture here. So technically, if these little squares, these diamonds, if we were following the rule, they go from a high to a low. There's definitely a higher concentration of them inside the cell than there is outside. So if it was passive transport diffusion, it would only be able to go this direction. Only this direction. But active transport can also go from a low concentration to a high concentration. So it can go both directions, low to high. Okay? Now you guys have this picture in your notes packet. Oh, how well you can see that. But you have this exact picture in your notes packet. So I want you to draw something for me. I'm going to move my face over here for a minute. But I want you to draw something for me. Let me see if I can get me my pen. So right here, this is actually a low concentration. Down here is a high concentration. Oh, all right, sorry, just a quick brief interruption. We had an announcement come on. So anyways, put a little low, and then you got high here. Okay, and that's what's unique, is that molecules can move from a low concentration to a high concentration and move this direction. But they also can go the normal direction. So we're going to put another arrow going this way. Because not only can they go low to high, they can definitely still go high to low. Me trying to make my arrow a little thicker. All right. Okay, so you just added that on your picture. It's right here. Is active transport can go low to high, or it can go high to low. The key thing is that it has to have ATP. It's got to have energy. Okay. All right, let's go on to the next slide. Okay, so now we're going to talk about bulk substances moving across the membrane. This is large, either molecules of water or of solids, doesn't matter. Okay, the key thing to know here is you can break up this word and it can help you. So endo means to enter. So endocytosis, this means to enter. Cyte means cell. And finally, acis means state. So if you put all that together, endocytosis is a state. The cell is in a state of entering. So things are coming into the cell. All right, phagocytosis. So you have the same words here, cell, state, but phago means eating. So now the cell is in a state of bringing stuff in, and it is solid molecules like your glucose, your amino acids, fatty acids. Okay? Now penocytosis, once again, the cell is in a state of drinking. So this is bringing in fluids, whether it's water molecules or any other types of fluids. Ooh, my writing is awesome. Regardless, it's bringing in big bulk of fluids, and that would be penocytosis. And then exocytosis is exactly what you're probably thinking, exit. So the cell is in a state of exiting. So now it's kicking it out of the cell. All right. So we're just going to talk about each one of these super quick. So endocytosis, once again, a process by which a cell surrounds and takes the material from its environment by engulfing them. So here's your cell, and there is whatever, if it's molecules, whether it's food molecules or liquid molecules, but it wraps around it. It's way too big to come in through a protein, and it can't diffuse through the cell membrane on its own. So it wraps around it and then brings it in, engulfs it. So let's just talk about this cell eating. This is a really cool process because, once again, eating. All right, the cell's in a state of eating. So the process in which phagocytes engulf and digest microorganisms and cellular debris. So this is like bacteria. Oh, here's my awesome writing with the mouse. This is like bacteria. Now this is a really, really important defense mechanism when it comes to fighting off infection. So if you have a white blood cell and it wants to kill a bacteria, it'll just go and wrap its body around it, bring it in. Then there's organelles called lysosomes, and they'll chop it up into little pieces. It'll keep whatever it could use to benefit it, and then it boots everything else out. And then that's what your kidneys and your liver help filter out and flush out of your body. So it's kind of really cool how it all connects together. So here's just a couple pictures. So once again, here is the cell. There's your nucleus, your nucleolus, and this is your white blood cell, and then here's a little bacteria. So it goes around the bacteria, it engulfs it. Now keep in mind this is two-dimensional, but it really is three-dimensional. It wraps it all up and brings it into the cell. So kind of an idea here. Now this video, I'm going to have to take my pointer off. Hold on one second. Let me pause this. All right, so here's the video I want to show you. Turn the sound on just a little bit.
Released by injured mammalian cells or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component.
All right, so the biggest thing here is happening is, once again, here's white blood cell, and it's going after this bacteria. And it just shows how it will actually connect up before it brings it in, wraps it up and brings it in.
The complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuum known as a vacuum.
So I love that it wraps it up completely. You can see that it's three-dimensional, brings it in. Don't worry about the names of phagocytes or any of that.
In the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digestive contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
So it kind of just gives you an idea how the whole thing works. It comes, it wraps it up, brings it in, lysosomes diffuse through, break it apart, and then it kicks out what it doesn't need. Really simple little process there. Pretty cool. Okay. Back to where we're at here. All right, so pinocytosis. Once again, the cell is in a state of drinking, so it's just bringing in large amounts of fluid. And same concept, wraps it around, brings it in, wraps it up in a vesicle, uses it what it needs. All right, so here's this one. Oh dear, we really got to stop this sound. And it stopped. So here comes basic fluids, bring it into a vesicle. And it just keeps repeating this whole process over and over. It kind of talks about, get a little bit further. Let's go to this spot right here because I like it. There you go. So here's that vesicle. Let me turn this volume on. And the cool thing is, you see how it reattaches back to the original cell membrane. So it brings it in. So once again, here is whatever the solute, if it's water or liquid, it brings it in, and then when it kicks it out, it reattaches back to its original membrane. So I want to go back to this right here, and we're going to pause it. All right, so I paused the wrong thing. Let's back this up right here and pause this video. So you can see, here is the phospholipid bilayer. Here's your two layers. This is what was wrapped up around the vesicle. So when it's kicking it out, the phospholipid bilayer just reattaches back to its original spot. So it's really cool how it does that. Okay, let's get out of this. All right, so that is pinocytosis. So phagocytosis and pinocytosis are both two processes bringing things in. And then you move on to exocytosis. Oh, we kind of looked at this already. Oh, so here's your live versions of phagocytosis. So here is, once again, an amoeba. There's one of your, and this isn't one of the cells in our body. Oh, all right. So we just had somebody pop on in, so I paused it real quick. Anyways, but when you look at these videos, this isn't a cell that's inside of our body. It's a different cell. But it is a single-celled organism swallowing up another single-celled organism. So it's really cool to see how it takes it and brings it in. Similar with this guy up here. This is an amoeba, single-celled organism, and it wraps up around it, brings it in, and then it slowly disappears. Really cool. Okay, so exocytosis is just a process when materials are transported out of the cell. So there's that phospholipid bilayer. It reattaches back up to the original cell membrane, and then poof, out the stuff goes it doesn't need. Okay, so it's really cool how it shows and it reattaches it back up there. All right, you have this picture similar to something like this in your notes packet. So what I just want to make sure is we notice is here's endocytosis bringing it in. It wraps it up in a vesicle. Lysosomes diffuse in. Right here. Lysosomes diffuse in. The food particles stay that they want to keep, and then the food particles they don't want stay in, and the stuff it wants to keep, it diffuses into the cell, and then we just kick out the rest. Okay. All right, so this is the picture that you have in your notes packet. Would you please, let's find, this is where it's going into the cell. So we're going to label this endocytosis because this is where it's coming in. And then over here, we're going to label it exocytosis. So I'm going to go ahead and get my, so we're going to write endo, and instead of writing the whole word, I'm just going to put endo. Then over here is exocytosis, where it's exiting. Now the fact that it's bacteria, we could actually even say, what type of endocytosis is it? Well, it's phagocytosis because it's bringing in food molecules. Okay? And then here's those lysosomes diffusing in to break it apart. The stuff it wants gets absorbed into the cell, and then what it doesn't want gets kicked on out. It's a really cool process. All right, so what I'd like you guys to do now is on this PowerPoint, you can jump on and do the diffusion little quiz. and see how you do there. All right, well, that finishes up active transport.